Non-clinical drug development process. In this process, the drug development preclinical trials are basically carried out by the laboratory test for new drug substances. Usually, these tests are performed by animal subjects to see whether the treatment really works or and it is safe to test on the humans. During the early non-clinical development process, the drug candidates needs to pass through several steps such as determination of the drug availability, which includes ADME, adsorption, distribution, metabolism, and elimination. These preliminary studies are aimed to investigate the drug candidate safety, which include genotoxicity, mutagenicity, safety, pharmacology, and general toxicology. The main goal of non-clinical studies, after identifying a compound, it is tested on the animals to expose the whole pharmacological profile. The main goal of non-clinical studies are determine a product safety profile to obtain the first information about the tolerability in different systems. Product may be include new medical devices, drugs, gene therapy solutions, etc. Experiments are generally performed on the rodents like mouse, rat, guinea pig, etc. After successful result, experiments are performed by larger animals like a cat, dog, monkey. After this process, completion of this process, the clinical trials are performed by the human volunteers. Some steps which are involved in the non-clinical drug development process, they are identifying the targeted drug, developing a bioassay, and screening the drug in assay, establishing the effective and toxic diseases. After that, finally, investigational new drug file can be prepared for the approval to start with the clinical trials. The objectives of non-clinical studies in drug development process. The purpose of preclinical study is to develop the data to decide safe human clinical trials on the drug of the drug. A laboratory test of new drug or new medical device usually done on the animal subjects to see the treatment which really works and it is safe to the humans. After completion of the animal testing, it performed by the humans. The types of study which involved in the non-clinical studies during drug development process, they are in this non-clinical studies process, some basic types of studies which are involved in the non-clinical studies, they are screening test on isolated organs, test on animal models, general observational test, confirmatory test, mechanism of action, systemic pharmacology, qualitative test, quantitative test, and pharmacokinetics. Last one is toxicology test. These are the tests which are performed by the non-clinical drug development process. Pharmacology study in the non-clinical drug development process. Pharmacology as an academic principle can be loosely defined as the study of effects of clinical chemical substances on the living organisms. This definition is so broad that encompasses all the aspects of the drug discovery. After screening, it involves the testing of drugs on isolated organs or tissues for detecting the activity like antihistaminic, antibacterials, etc. Test of the drug candidates on the animal models which are induced with the human diseases is also done by the drug efficacy on the disease. The mechanism of action of the drug candidate is also determined in the section of the study. For example, antihypertensive is whether calcium channel blocker or beta blockers, AC inhibitors, etc. For antihypertensive patient, these types of drugs can be given irrespective of the primary action of the drug. Here we also study about the systemic pharmacology and effect of drug candidate organ system like such involves nervous system, cardiovascular system, renal system, respiratory system, etc. After that, pharmacokinetics like drug metabolism studies in non-clinical drug development process. The majority of pharmacokinetic studies involve the measurement of specific compound in easily sampled physiological fluids like blood, plasma, serum, and saliva. These excreted substances are also measured in the urine or feces. In some rare situations, sampling techniques like tissue biopsy, cerebrospinal fluid, bronchovascular fluid can be taken for the test processes. The central role of pharmacokinetics in drug discovery to contribute the compounds for man by the balancing of the properties. By these pharmacokinetic studies yields the parameters such as volume of distribution, half-life, mean residence time, area under the curve, maximum concentration of the drug in the blood, and time of the Cmax. It can be studied out by the pharmacokinetic studies. The dose response relationship, maximal effects, comparative efficiency, drug dispersion, these these studies can be studied by the pharmacokinetic parameters. After that, toxicology study in the non-clinical drug development process. Non-clinical testing contains the bioactivity, safety, efficacy of the formulating drug product. In In preclinical toxicology studies, it play an essential role for providing the clinical studies to determine the drug compound. The primary objective of the toxicology studies in the drug development process by using the relevant animal models and validated procedures on the animal model responses to undertaking the risk of human or subjects. During this preclinical stage, the development process and clinical trials of the investigative new drug applications are prepared. Some different types of stages which are carried in the toxicology studies, they are acute studies, repeated dose studies, genetic toxicity studies, reproductive toxicity studies, toxicokinetic studies. In acute studies, single dose of the drug will be given on the animals for after dosing that 50% of the animals is called as LD50. After depending upon the duration, the studies can be repeated by the dose studies. The specific duration should be anticipate the length of the clinical trials. These studies assess the likelihood that the drug compound is mutagenetic or carcinogenic. After procedures such as AMES test detect genetic changes by the DNA damage in testing. These tests can be carried out by the gene toxicity studies. Basically, in this study, the segment 1 reproductive toxicity studies of the drug effects on the fertility. In the segment 2 and 3 studies, the embryonic and postnatal development defects can be seen. After that, in general, reproductive toxicity studies must be completed before a drug can be administered to the women of the childbearing age. These are typically similar to the pharmacokinetic studies, whereas they examine the effects of toxic doses of the drug, which helps to the clinical margin of the safety. Another topic is investigational new drug applications. Approval to the investigational new drug applications allows a company to initiate and conduct the clinical studies for new drug products. In these new investigational new drug application provides data necessary to decide the new drug and preclinical maintenance, which is a reasonable risk to the human subjects.